describing the work that we've done under the Sustainable Seas project, I'll try and answer the questions that were asked. And the first question that was asked was the most recent algal blooms, that's over the last 2 years in the Sounds, closing the area's harvest, are they different from what has occurred in the past? Well, yes and no. The main cause of these blooms has been a dinoflagellate known as Alexandrium pacificum, and it was first observed in Queen Charlotte Sound in 2011, and then appeared in Pelorus Sound in May of 2018. We've known that this particular species has been present in New Zealand for a long time. It's actually been particularly common on the east coast of the North Island, from the Bay of Plenty northwards. In 2017, it extended its range down the west coast of the North Island, and then subsequently appeared in the Marlborough Sounds. The reason for the problem is that this particular dinoflagellate produces paralytic shellfish toxins. And I won't dwell any more on that aspect of this thing, but suffice to say that they are very potent neurotoxins. They potentially fatal in high doses, but there's very strict internationally mandated levels that are permitted in shellfish throughout the world. And so it is absolutely crucial that any products coming from New Zealand do not contain these compounds. And over the last couple of years, it has caused prolonged shellfish harvest in Pelorus Sound. It can cause, under extreme conditions, discoloration of the water, so-called red tides. A couple of examples seen here in the Marlborough Sounds in the last couple of years. And the figure on the left here is just shows how Pelorus Sound between May and June 2018 essentially got measles, and the red dots show the relative distribution of the dinoflagellate in various other parts of the Sounds, essentially closing the whole entire area. But from that figure, you can obviously see where the most intense bloom was, and that was in Nydia Bay. In the picture, the aerial picture to the right there, Nydia Bay is that inlet going off to the left here as you look out northwards down Pelorus Sound. This figure here shows the levels of paralytic shellfish poison in mussels this year, from between December and July of this year. The low levels that are seen in December are actually residues from the year before, still existing in the mussels in Nydia Bay, in that area. And as you can see from that figure, that the main focus is in the central, whoops, sorry, that wasn't supposed to happen. How do I go back? No. Is mainly in this central area here, focused on Nydia Bay and Pelorus Sound proper. There seems to be another area over in the western side of the Sound too, where the bloom has been generated essentially at the same time. This is a figure showing the abundance of the dinoflagellate in the water column and associated paralytic shellfish poison concentrations in mussels from one of the monitoring sites in Nydia Bay. As you can see, that the toxicity lags somewhat behind the dinoflagellate cell numbers. The second question that was asked was, are these events likely to become more frequent now that they've become established in the area? And I think we can say absolutely yes, that is absolutely the case. And the reason is that this dinoflagellate, similar to some other dinoflagellates, has a benthic stage in its life cycle. So that's the diploid stage of the life cycle, down the bottom there, down here. And when conditions are right. Exactly what those conditions are, we're not totally sure. Oops, that's very sensitive. And then the cysts germinate, the motile form of the dinoflagellate goes up into the water column, and then it simply divides by binary fission until such time as it receives some environmental cue. Again, exactly what that is not terribly clear. Dogma would have it that it's nutrient limitation, but we've found that that's not the case in Pelorus Sound. Anyway, two different mating types of cells get together. They produce a zygote which swims to the bottom, sheds its cell wall, and insists in the sediments on the sea floor. And so once a seed bed like this is established, it will continue to generate new blooms annually. And this figure here down the bottom there is actually from a survey we did back in August in 2018, looking at the numbers of dinoflagellate cysts in the sediment, and in particular cysts of Alexandria pacificum. There are high numbers of cysts in the sediments there, some several millions of cells per square metre of sediment in the top centimetre. And so undoubtedly new blooms will be generated from that area now. There was another question, was that what was that question here? Oh yeah, do we think the occurrence of these types of events will increase over time? That's a rather difficult thing to answer. But we have an example in Queen Charlotte Sound where the dinoflagellate appeared to arrive somewhere back in the 2010 time. And since that time, and established, there was major blooms throughout Queen Charlotte Sound, closed all the shellfish industry in that area for some time, and established very high density cyst beds in Opua Bay, which is an inlet off Tory Channel. But since that time, actually, it seems to have been winding down. The blooms have become much less intense, and their duration is reduced. And it's all about, we think, what happens to those cysts on the seed bed. We're very lucky in a way in this year during midwinter, when the figure down the bottom shows that figure I showed you before. And you see the dip there and before the major peak. Essentially at that stage it looked like the bloom had completely gone away. The photographs at the top, they're satellite images from the Sentinel satellite. And there was a period there, you'll notice the timing of this. This was right over the winter solstice. It's the time when the sun's at its lowest in the sky, the days are shortest, the water temperatures are as cold as they get, about 11 degrees. And to all intents and purposes, it had appeared at that time that the bloom had actually collapsed, but in fact it hadn't. It had simply disappeared from the monitoring sites. And one of the other questions that were asked was, from a commercial point of view, what benefit does early detection of blooms provide for the mussel farmers? Well, I think it's pretty obvious from this picture, is that they could have easily been caught out here if they hadn't resumed harvesting in Nydia Bay at that time, but they could have, and within a period of 5 days, the bloom had been generated in the top of the bay and then dissipated and contaminated all the farms in the inlet. So how do we monitor for toxic phytoplankton? It's generally a very simple process of looking at samples under the microscope with a rogues gallery of things that we're looking for. And what we've been trying to do under the Sustainable Seas project is to explore some other options to try and provide methods that are quicker, simple, quick methods that are perhaps cheaper and can give us greater spatial and temporal coverage of water bodies like the Sounds. And the first one is a portable DNA qPCR system, which you see here in this picture. It involves a technology known as Hydrogel qPCR. The magic is in the tubes in the bottom, where all the qPCR reagents are contained in a gel, and it's simply a matter of taking a water sample, filtering it through a small filter, extracting the DNA in the shaker, putting the extract into the tubes and running it on the thermocycler, which is the red instrument there. And it certainly works. It's a sort of thing that you can use on a small boat. You can have the results of about 16 samples available in about 90 minutes. And in terms of time and labour, it's really very competitive with conventional microscopy. The trouble is that the reaction gels that we were using actually weren't all that good, and unfortunately the business that was the Canadian outfit that was actually making these gels has subsequently gone out of business, so we can't access them anymore, and their reliability wasn't that great. They did work, the sensitivity was very good, but the reliability and the precision of the quantification really wasn't good enough for regulatory purposes, especially where it's a food product is involved. And so we haven't given up on this. We certainly won't use the hydrogels again, but there are numerous other options to achieve the same result. The other technology that we've been trialling is an instrument known as the imaging flow cytobot. It's essentially an autonomous submersible microscope. It's made by McLean Labs in the USA. It was developed at Woods Hole Oceanographic Institute in the States, and then they've sold the license, the patents to McLean Lab, who now manufacture them. They're specifically designed for identification of and counting of phytoplankton. And for the last nearly 9 months now, we've had one of these instruments on loan from Professor Rafe Kudella, University of California at Santa Cruz, and we've been trialing this instrument. This is images off the McLean website. You can look up their website and see a video of how the instrument works. So I've taken a few frame grabs off that. And it simply acquires a water sample by sucking it up with a syringe, and then the syringe expels the sample through the cell, the counting cell. It has quite an interesting feature, which is different from other flow cytometers, is that the actual sheath fluid that's used is actually the sample that's been filtered. Which is actually a really good feature. It means that it can be left unattended for long periods of time, continuously sampling. And there have been deployments of these for 6 months or more, continuously sampling. I think one of the good technical aspects of that is that because the refractive index of the sample is the same as the sheath fluid, it gives really good focus and sharp images. Anyhow, the flow goes through the flow cell, laser beam. As the particles flow through, the light is scattered from the laser beam. It also induces fluorescence in the cell if they've got chlorophyll. And the machine can be triggered to take images either from scattering or from chlorophyll fluorescence. In scattering, you get everything. You get every particle that's in the water. With chlorophyll, you only get chlorophyll-containing particles, and which is a huge advantage because you can put a water sample that contains a lot of inorganic detritus and stuff, silt, and through, and you still get really sharp images of phytoplankton. So we've deployed it in a couple of ways, one on our small research vessel, seen there on the left. And also we did a 3-week deployment on New Zealand King salmon farm out in Pelorus Sound at Kapua. This is the sort of raw data you get from the instrument. It takes 20 minutes to run a sample, a 5 ml sample, and it will count and identify, but take a photograph of every particle that goes past the camera. And so you end up with these images. And it's easy to recognize, you know, what is in those generally. The one on the lower left actually is quite an interesting example. It was just actually taken last week from Nydia Bay. A phytoplankton community dominated by diatoms, as is normal for this time of year during the spring bloom. But down in the lower right-hand corner, there's a couple of dark stains, not stained, but dark coloured cells. And these are the first signs of Alexandrium pacificum reappearing in Nydia Bay for the next season. So as you can see, it really is quite obvious to pick these out. We had another problem this year, though, that as I said, Alexandrium pacificum has just appeared in Pelorus Sound in the last couple of years. Well, this year, another species, Alexandrium fraterculus, which is the cells on the bottom of this image, which actually happens to be non-toxic, that also appears in the Pelorus Sound. We've never seen this before. It's the only time, to my knowledge, that we've seen it before is in the Hauraki Gulf. Somehow it's made its way down into Marlborough Sounds. But distinguishing between these is a bit of a challenge. And it will be a bit of a challenge for the image classifiers that can automatically identify the species in there. And when it's easy, you can see when you look at these cells that they are, although they're very similar, they do have slightly different shapes. They're a similar size. One of the outstanding features of Alexandrium fraterculus is the fact that it forms these long chains. But if you're getting the ventral or dorsal view of the cells, you can tell. But if you see them in any other aspect, they're just ambiguous and you cannot really tell what they are. This is the result of the deployment off the King Salmon's farm in Pelorus Sound. Over 3 weeks, nearly 1,000 sampling occasions, 600 odd cells of pacificum seen, about 300 specimens of fraterculus, and about roughly close to 30% of the cells were classified as ambiguous. This sort of data is completely impossible by any other means apart from this. It would be totally impossible to do this manually, that sort of volume thing. The other information that the instrument gives you, which is really quite incredible, is that it gives you the whole community. It shows you everything that's in there. And that really does have a bearing on the dynamics of bloom, something that we know is important, but because it's so difficult to do manually, we can't do that. So I won't bore you with the details of that, except to say that although Alexandrium pacificum was common there and the mussels in the vicinity were contaminated, it was a very, very tiny proportion of the phytoplankton community. There's good quantitative relationship between estimates of abundance manually and by the site of both manually identifying from the photographs and also being put through the artificial intelligence cell classifier. And other information that the instrument gives you is things like very intimate views of what's actually going on. In this case, the cells at the bottom here, Dinophysa acuminata, produce diuretic shellfish toxins. It's a mixotrophic dinoflagellate. It actually feeds on the ciliate, the Mesodynium rubrum. And down the bottom, you can see cells that have just had a feed of that. And that's an indication of, once they've had a feed of Mesodynium, they multiply very rapidly. So it's a kind of early warning thing. Phytoplankton that are hazardous fish are detected by the cytobot. There's quite a number of targets there that are certainly hazardous to fish. The one right down the bottom, and the other thing that it gives you, you see things that although I've spent my lifetime pretty much looking at phytoplankton water samples that I've never seen before, and seen things that I thought were rare and actually turn out to be really rather abundant. So it's quite good for some of the fish killing species. However, it's less successful some of the small species such as Pseudochatonella and Chrysochromiolina, but this applies to manual microscopy as well. So pro-cons of the machine from the work we've done on it. It can give real-time enumeration of the harmful species. It's really good at that. It gives us whole community analysis, which is fantastic. So we can understand bloom dynamics better. The data is of really good quality. One thing I didn't mention, you can get much higher sensitivity than is possible through microscopy by pre-concentrating samples. You've got a permanent record of all the samples you've looked at. You've got this ability to do in situ observations and have it remotely deployed and transmitting data back to the lab. Gives you automated size and volume sorting data. It's powered by open access software, so there's no expensive software updates. It can save an awful lot of labour. And this instrument is the current state of the art. It's not perfect. If I was to wish for a redesign, there'd be a number of things I'd have. I'd say it should have more than one channel. It should have several channels. Perhaps be better resolution for the very small cells. And it doesn't really, for an instrument like this, we really don't need it to be placed in the water. So we don't need all the pressure housing and all that sort of thing. But some of the slight downsides of it is there is this ambiguity between species. It's sometimes hard to tell exactly what it is. Smaller species are not well resolved. Some of the large species, such as Pseudonychia, are not quantitatively sampled very well either. They're just simply too large to get into the sampling tube. It requires live samples in good condition, and that's a bit of an issue when you have to transport samples from, say, the Marlborough Sounds to a laboratory somewhere. And the 5 mil limit is okay. You can run samples in duplicate to increase the sensitivity, and 20 minutes isn't too bad, but you can only do one at a time, of course. So in summary, the imaging flow cytobot can enable this real-time sampling analysis, greater spatial coverage with reduced labour. It hugely enriches the quality of the data collected. It's a really important research tool. Can provide early warning, optimise shellfish harvesting, protect public health and ensure market access for aquaculture products, which after all is what this is all about. There's still work we need to do to customise the image analysis classifier to New Zealand species that we're encountering here. It could be introduced now to supplement the current shellfish toxin program and would really make a useful addition to that. And but molecular approaches may still be more suitable for screening for some of the small fish killing species. And that's it. Thank you.
Try out the mouse because I saw Lincoln was having a few problems with that. Yeah, good afternoon, everyone. I'm Ben Knight. Welcome to our audience here and the cyber audience. Just a reminder, there'll be questions at the end and you can add those to the chat. So today I'm going to be speaking about near real-time forecasting simulations that we've been running for Tasman and Golden Bay with colleagues from NIWA, David Plew and MetOcean, Brett Beamsley leading the team there. So this has been a combined effort between NIWA, Cawthron and MetOcean Solutions. This sort of fits in with the wider Sustainable Seas Challenge. Its vision to provide healthy marine ecosystems that provide value for all New Zealanders. And digging into the valuable seas component that this program sits within, the bit where this is focused is really looking at identifying innovative ways to add value to the marine economy. And really, by marine economy, we're looking at aquaculture here and also marine tourism, so the use of the beautiful beaches we have around the Tasman and Golden Bay region. A little bit about my background. So I'm an ocean swimmer as well as an oceanographer, so I like studying the water, but I also like getting in it. So this work has direct relevance for me, hopefully in the future. A bit more background, why are we interested in it? A lot of people know about sort of the upset bellies and stuff you can get from swimming with bacterially contaminated water, but you can also get some quite nasty things. And I did a bit of a scour of the internet for the worst, the worst possible examples, and found things like flesh-eating bacteria and man dies from tattoos. So that's another important point, that your contact with bacteria is not just coming in through ingesting it. Potentially, if you've got cuts on your feet, you can have problems and things like that. So there's a lot of sort of safety aspects to consider. And in terms of shellfish, I mean, just recently we've had the recent Vibrio scare up in the Coromandel, which left some people in hospital. So yeah, some of these bacterial issues can be serious food safety issues. So on the top of the south, I think this is a really nice image because it shows a few different things about the bay. One is obviously we get, after large storm events, we can get a lot of sediment and bacteria pushed into the bays. And you can see these little white boxes represent the consented areas for aquaculture in Tasman and Golden Bays. And potentially this is going to be very significant in future. So about a third of New Zealand's shellfish production could come out of this area in future. It's not there yet, but you can see that there's serious risks for an industry which is trying to develop here and is also exposed to these sorts of events. So really what we're trying to do is give that industry better information on how they might harvest. So you could see, for instance, some of these farms are being affected by rivers and some of them are potentially not so affected. So being able to choose which farms can be harvested and have that sort of greater spatial resolution and your decision making is going to be really valuable in future. For those of you that don't know what a mussel farm looks like, I just thought this would be a useful slide. So if you're sort of sitting on a boat on the water, you can see that all you can see is these little buoys sitting there. But when you get underneath, you can see that underneath those buoys, you've got these lines or ropes with mussels hanging on, and each one of those mussels potentially can filter several litres of water per hour. So they can accumulate the bacteria that's in the water, and like Lincoln was presenting, toxins potentially. So yeah, I mean, this is a really important issue for this species. And of course, everyone knows about the beaches around the Golden Bay, Tasman region, and this is just a picture of a beach from the Able Tasman. So obviously a very important tourism area, and giving tourists information about when it's safe to swim could save a lot of negative news stories in future. So just to give you a bit of background about where we're focusing, obviously there's lots of different sources of bacteria. For this work, the study, we've focused particularly on the diffuse inputs, so those coming off land and down the rivers. But of course there are point source discharge as well, which is sewer overflows, stormwater. Those other sort of inputs are not considered in this study. And that I'll discuss that a bit at the end, but that might have some implications for where we can predict reliably with this model. Another important point is that we're just looking at indicator bacteria here, so not necessarily the things that make you sick, but the things that are indicators of those other potentially harmful bacteria. So the two species we look at in New Zealand in our regulatory system is E. coli, and that's mainly for freshwater, but also applies to shellfish harvesting, and enterococci is the other one. And the reason we use enterococci in the marine environment is that it's a bit more robust, it's able to survive in that saltwater environment a bit longer, so it's a better indicator of sort of far field effects of these rivers. So what does our solution look like? I'm a modeler, and the people I'm working with are computer modelers, so we think a modeling solution was the most practical in this to achieve what we wanted to achieve for this project. And you can see it's a bit of a horrendogram here, but just breaking it down a little bit, we've got a kind of a land-based modeling exercise going on the left, and so what's happening there is we're running a meteorological model, and that's basically providing rain forecasts, which then feed into a catchment model, which then provides river forecasts, and NIWA's led that component of the work. And then on the other side, we've got a coastal transport process going on. That's led by MetOcean, who developed the coastal models there. And as well as the coastal models, they also require things like wind and rain and solar radiation to drive them. So they do also require atmospheric inputs into them. And basically, once we have all that information, we're able to estimate bacterial concentrations in the river and combine them with these transport models to produce outputs, which I'll go on to show now. So just to give you a bit of an idea of what those weather models look like, we're all used to seeing weather models. So this project is basically feeding those rain forecasts into a river forecast. So we have some predictive ability with these simulations. And we're doing these for 12 different rivers in the region. So you can see there's a map of most of them there. The colours here indicate the size of the rivers. So there's a few major rivers like the Motueka in the middle here, also the Takaka River over here, and the Arariri. They're sort of the big three for the regions. Also the Waimea River here, and then lots of sort of smaller catchments and rivers on the Nelson side of the bay. Interestingly, I'm not sure how many of you are familiar with this River Maps website, but I just wanted to give them a bit of advertising there. It's a product that NIWA's produced that has a lot of information on rivers, which I've found really valuable. So if you look down at the bottom of the screen there, I've got a link to their website. But if you just search for NZ River Maps, hopefully you can find it. So in terms of once we have the river flow information, we need to estimate river pollution. In the last talk I gave, I had quite a few questions about this, so I thought I'll just throw in a slide that gives people a bit more background about how we're estimating E. coli concentrations in the rivers. So we're really lucky in the Nelson region. We have a really good state of the environment monitoring program going on for the major rivers, run by Tasman District Council. And they're sampling regularly through the summer months. In order to be able to predict models, what we need to know is what's happening around flood time. So that required us to set up some to actually go into the rivers and take regular samples through a flood event. So we've got a lot more information from our own targeted sampling as well. And we've managed to build some models for the major rivers that are quite successful. So you can see the number of data points here for these major rivers and the sort of performance we're getting. The only one that we've really struggled with is the Waimea River, and that was one where we didn't have that targeted sampling around floods. So potentially something we can improve in future. For the other 7 other rivers that are a lot smaller, we've just used the data that's available on the NZ River Map tool to estimate those. So potentially, when this model's running in future, we want to improve it for particular areas. We can go back and do some targeted sampling around those smaller rivers and improve our estimates there. So the coastal transport model is a high resolution hydrodynamic model. And you can see here that it's basically formed from a bunch of small triangles, and those triangles vary in size. As you get close to shore, they become a lot smaller, so we have greater resolution. And this is really important if we want to predict what's happening at individual beaches, which some of those beaches can be quite small. Just to give you a bit of a zoomed in view, this is the Waimea estuary. You've got Nelson over here, Tahuna Beach. You can see that there's a lot of detail in these models. This is models that MetOcean have produced. You can see they're able to model individual channels, which is really good if we're wanting to find out information for a specific site. In terms of the bacterial transport, once it gets into these coastal models, it basically starts to die off. The mechanics around it are pretty complicated, so it's affected by the salinity in the water, the light intensity of the light, so UV will kill it off, and also the shading, so things like sediment in the water. So we've had to simplify things somewhat, so we're not modelling sediment, so we can't estimate those sorts of things. And so we've just taken a simple approach of assuming a constant die-off depending on the time of bacteria. You remember I said earlier that the E. coli tended to die off earlier, so we've got a time for 90% die-off of 24 hours for E. coli and 48 hours for enterococci used in the model. Once we have all this information, we can then start to do forecasts. So this is actually a system that's up and running that you can log in to now. It's a portal provided by MetOcean. And if you log into MetOcean View, you'll have to set yourself up with an account to get access to this. But basically it'll enable you to look at what's happening in those bays at the moment, and also a day into the future. So you can imagine for a mussel farm that's wanting to plan out the next day's harvesting, there's been some rain, they might want to go and consult this first before they harvest product that potentially could be contaminated. So you can see there's a lot of value in having a bit of a forecasting ability from this system. Just to give you an update of what it looked like yesterday, you can see that we had a bit of rain recently, and for instance, the Motueka River plume was potentially some contamination affecting some of the mussel farms off the Motueka River. So you could see that some of those sites would be affected, but potentially there were areas which weren't exposed to very high concentrations of bacteria. So by having this fine spatial information, we're hoping that we can improve access to the non-contaminated product for aquaculture in the region. Just to give you a bit of an idea how that flow peaked, so we're looking at sort of 300 cubics in the Motueka. So that's about 3 days before this image was taken. So potentially, even with that die-off, still a bit of persistence of bacteria in that system. In terms of making this information more accessible for swimmers, we've also sort of prototyped a system that can hook into the model data that's provided in that more sort of technical format for mussel farmers, and it can provide sort of simple swim, no swim information, and that can say potentially be picked up and improved by council, just to get that information out to people so they are aware of the potential risks of swimming after rainfall. So just to I've presented a lot about what this model's done, but yeah, a model without data is really just an opinion. So we have done a lot of checking of things like the hydrodynamics, how the water moves in these models, and that's turned out pretty well, and also sea surface temperatures able to be incorporated into these models if that was used in the die-off information, potentially we could incorporate that in the future. We've also got a lot of access to bacterial information that's been collected over the years from the aquaculture industry. They've been really generous with their data, and also the council bathing water data, state of the environment data that's collected around the bays. So we have done, we've also done some targeted collection of our own. So assessing the performance, we're still going through this at the moment, so I can't give kind of a definitive statement on the performance of the models yet. It looks like some sites are doing okay, other sites, potentially there's some effects from non-diffuse sources that could be influencing those. But basically, yeah, we've gone back for a year, run the model, and then we're able to compare it to the data that we've collected there. Yeah, and we're just in the process of trying to figure out, yeah, the performance and get that all sorted. So in summary, yeah, we can see that bacterial pollution is a serious problem. These types of tools offer the ability to better understand where it's coming from and how we can manage around it. We're still assessing the performance of the model, so I can't give you a definitive statement that it's perfect yet. And but hopefully it'll lead to cleaner water in the future, because I think when people are aware of potentially where the issues with their local swimming feature coming from, they can start to talk to the council about trying to improve and clean up those particular catchments. Yeah, if you have any information, feel free to get in contact with me. My email address is there. And thanks for listening. Just a reminder that Robin's going to come up and coordinate questions.
Wow.
Oh, sorry. Did you want to come up as well, Link? Or?
No. Thank you very much, Lincoln and Ben. I would just say that, yes, if you are joining us virtually, please submit your questions via the chat. If you sort of hover over at the bottom of your screen or at the top, depending on where your kind of little Zoom green and red bar is, there's a chat function there. You can click on it, send the question to everybody, and then I can read it out to these guys. In the meantime, is there anyone in the room? And if you two could repeat the question just to make sure that everybody has heard it, because the mic's directional.
Sure. Okay.
Anyone in the room? We do have one from Robert Shepard. I can't pronounce the algal. It's the Akashiwo. A hub with problems for wildlife. Is this part of monitoring as well? The Bradenberg 2018 meta-analysis reveals enhanced growth of marine harmful algae from temperate regions. Our results warn for a greater potential of dinoflagellate HAB development in future coastal waters, particularly in temperate regions. Would you agree? So, Lincoln, if you said that way, you're in. Sorry, try and repeat that question.
So I think the gist of the question was that is there greater likelihood of increased dinoflagellate blooms with global warming, I assume? Maybe. The one rule of thumb that dinoflagellates, of the conditions that dinoflagellates really like, is a stratified water column. That's the case with the situation in Nia Bay. And the reason for stratification there, which persists right through the winter, is because of the freshwater inputs from the Pelorus River and the stratification that develops in that bay, which is pretty stable. Whereas in Tory Channel and in Ipua Bay, stratification there is all thermal pretty much, because there's very little fresh water. So hence the blooms are occur in the summer there, whereas they occur in the winter in Pelorus Sound. Any warming of the seas possibly could lead to more intense thermal stratification of the water column, which would enhance the habitat for flagellate species, including toxic dinoflagellates. But as I tried to point out, not very successfully, that often the actual toxic forms are just a very small proportion of the community as a whole. In fact, Alexandrium pacificum seems to be a very poor competitor with other species, and just because there might be increased growth of flagellates in the water doesn't mean to say there's going to be more problems with intoxicated shellfish or effects on wildlife.
Got one from Stephen Gray, which is, how significant is sediment, especially from pine plantations, in the life cycle of these algal species? This is from Friends of the Mata. So, sorry if you could repeat that again.
Yes, I think the question was, how important is sediment from runoff from forestry operations in the Sounds? Certainly there's, when you see some of the satellite images of sediment plumes coming down the Pelorus and off the various hillsides in Pelorus, the sediment inputs can be pretty significant. However, whether that really has any bearing on dinoflagellate blooms is questionable, really. In fact, there is in Korea where they have quite significant problems with algal blooms affecting fish culture. Their major mitigation method is actually the spraying of clay in the water to flocculate blooms. And it's actually quite effective. They can actually protect fish farms by spreading clay around. So, no, not really.
I'll throw it in there as a non-technical person. It's possibly a bit basic, but if I was a regional council or someone from an aquaculture company that was looking to kind of get these things on board, what sort of time frame and level of investment, or how far away are these things? That's probably applies to both of you, so maybe Ben and then Lincoln.
Yep, sure.
Repeat the question if that's okay.
Okay, so Robin was just asking us if sort of how close are these tools that we've been investigating, sort of how close are they to being ready for market, basically. So I think in the case of my study, so there's obviously some performance assessments that need to go on to check how good the models are working and making sure that they're fit for purpose. Once that work's complete, the models are essentially running every day already. So there may be some tweaks we need to do to make them sort of fit for purpose. But yeah, I mean, within the next few months, hopefully we'll have something that's ready to test. And I think because there's a human health aspect to my modelling work, there's probably a certain gaining of confidence that would need to occur. It's all very well to sort of check your model in the past, but you're wanting to check that it's going to work into the future as well. So I think the probably a cautious approach to implementing it is needed for our work. Yeah, but essentially all the tools are there.
That actually, it ties nicely with the question that came in as I was talking, which was, are there mussel farmers in Tasman and Golden Bays using these models now to make decisions about harvesting? So have you got people lined up to help do that validation? Yes,
yeah. So we just had a question come in that was asking, are mussel farmers using it already? And the answer is no, we're still in a development stage at the moment. They've provided us all their historic data so that we can do some checks of how the model compared to their historic data, but those checks are still working. So we won't sort of go live with the mussel farmers until we've done those checks. But potentially some of the information is still useful in the absence of any information. So if they see the model showing a big bacteria plume affecting their farms as we've predicted, they may potentially make the decision that it's better not to harvest, or they might sort of take water samples in different parts of the farm that appear to be unaffected by the model results, and be able to check whether, hey, potentially we could look to harvest it if our measurements come back safe. So I think there's potential to use it even while we're still in the sort of validating stage, but yeah, it's definitely a possibility. I'll pass on to Lincoln because I think that first question from Robin applied to him as well.
So that was how would they be applied? I think that was the question.
Someone wanted to do that.
Somebody wanted to do it. With the molecular methods that I described, it's perfectly feasible for those to be run, say, on a salmon farm, or even on a mussel harvester, really. It's very compact, and as long as you have a power source to power your laptop and the thermocycler, it's very simple. It requires a little bit of rigor, and there's a couple of pipetting steps that you've got to get right. But if we had a stable, accurate system, which the hydrogels initially promised, but really didn't live up to their promise, yeah, it would be quite feasible to actually do it on site rapidly. There's always be an issue though with these qPCR assays, and that's really around the precision, because there are question marks as to whether the gene copy numbers in cells are constant or not, and there's various options for variability. So perhaps for food safety things like actual paralytic shellfish poisoning, you do want high precision because it's a food safety issue. But for things like protecting your farm from fish killing, you don't really need that level of precision. You want to know if it's there or it's not there. You want to know whether there's a little, some, or a lot. And that's really very feasible. As far as the Cytobot goes, well, that's a $250,000 instrument, and I don't know. I think it would be. It's a step on the path to the holy grail of having small, cheap, in situ instruments that are automatically telling you what's in the water and giving you an alert. But this is still essentially at the large prototype stage, and maybe that'll come in time. But at the moment, an instrument like that really, I suppose, will have to be in an institution like the Cawthron or NIWA or some other laboratory services.
Thank you. I think we have time for one more question, which is Tanya Gray. I think it might be for you, Ben. It says, you say that your model doesn't account for point sources. However, if there's a sewage spill, would it be useful to be ready to monitor being intensely so we can build this into the model application? So, feedback.
Yeah, so I just had a question come through that has asked me about the importance of, or is it possible to include point source information in the model, particularly if they know particular events have occurred, so some sort of sewage spill event. And yes, it's definitely feasible to include point sources. It's really just getting access to the information to be able to put into our models. So yeah, I mean, the framework's here now, so it's definitely something that we can build on, and something we'd like to build on, particularly for the more sort of urban areas in the Tasman-Golden Bay region. But in terms of the focus for this project, because it was such a aquaculture focus, and a lot of the beaches are sort of in rural areas, we kind of made the decision that diffuse was the best for the initial focus for this project. But yeah, I think in future, adding that capability of point source events as well would be really valuable.